Welcome to the final presentation for the day. To provide a snapshot of what we have been talking so far and to provide suggestions regarding how to combine it all, the focus of this talk will be risk assessment and decision-making process in primary production. So, frequent involvement of foodborne pathogens in fresh produce linked outbreaks led authorities to provide scientific opinion and technical guidelines as part of their risk communication plan. They published several documents related to microbial risk involved with fresh produce, including lettuce, tomato, berries, melons, all the foods that were involved in the fresh produce linked outbreaks. So, what are the commonality between those documents? What are the key messages? When we look at those documents, the microbial safety of fresh produce depends on three key factors. And those are, initially, to identify the main risk factors for specific food and pathogen combinations, including agricultural production systems, origin and further processing. Secondly, to recommend possible specific mitigation options and to assess their effectiveness and efficacy to reduce the risk for humans posed by these food pathogen combinations. And third, to recommend microbiological criteria, if relevant, for the identified specific food pathogen combinations throughout the product chain. So, the law that describes the general hygiene requirement for all food business operators requires the implementation of HACCP for effective monitoring of the risk. However, in primary production, the implementation of HACCP is not usually possible due to the difficulties associated with the identification of real critical control points. It's merely because HACCP requires you to identify these potential hazards and then determine the significance of these hazards by applying risk assessment techniques, the output of which is a measure of risk, which then allows us to put in place appropriate control measures. So the question is, what methodology should we follow to manage hazards in primary production? So to this end, WHO and FAO suggested a risk analysis framework that enables an enhanced understanding of the food safety and management of hazards in primary production. So the risk analysis is a process consists of three interconnected components, and these components are risk assessment, risk management, and risk communication. For risk assessment, it's defined as a scientific process that aims to quantify the risk, which is defined as the probability and severity of an adverse health effect consequential to a hazard present in the food. The risk management involves careful evaluation of the contents of the risk assessment, while the risk communication is the process where interactive exchange of information throughout all relevant stakeholders during the risk analysis framework take place. So when it comes to risk assessment approaches, there are two different approaches in estimating the risk. The first one is bottom-up risk assessment, and the hazard is the starting point, and it's followed by a consideration of all events that affects microbial load across the value chain to produce a prediction of risk to human health. On the other hand, the second approach, known as a top-up risk assessment, it involves the starting point for this approach, it's risk itself, and epidemiological data, it's the baseline to make a comment about the hazard. And most of the time, the availability of the data makes it hard to apply top-up risk assessment. In an ideal world, validation of each assessment approach should be aimed to have a more realistic overview of the risk events. So, as part of the bottom-up risk assessment, quantitative microbial risk assessment has been used to estimate risk and consequences from an exposure of individuals or populations to infectious microorganisms. It identifies magnitude of risk and provides option to evaluate intervention strategies to eliminate risk. So once you identify which hazards in primary production you have the potential to cause an adverse health effect, you need to apply exposure assessment, in which you need to define what is the intake of the hazard throughout the food. And this includes initial concentration, prevalence, and also chances for cross-contamination. Therefore, to give an accurate output on exposure assessment, we need lots of data. So how do we approach it? What we need to do is compile all the data relevant to primary production, and this includes industry data, experimental data, local data, and literature data. We need to use all this data, and of course, we need some science-based credible assumptions to make a comment about how produce gets contaminated and how to manage the risk. So to this end, what approach did we follow? We looked at the known parameters that we have, and then our aim was to mimic Australian primary production conditions to assess its microbial risk for the leafy greens. We developed a risk model that consists of 5 modules, in which all key events that affect microbial load were considered. And as an indicator microorganisms, we used generic E. coli data and transform it to the pathogenic E. coli by using credible assumptions from the literature. We compile all this data and use it to make a comment about the things that we don't know about the process and how to produce get contaminated and how can we manage the risk. So as a black dotted line represents here, that is the baseline for summer harvest. And the mean pathogenic E. coli levels under Australian conditions for summer season varied from 0.6 to -3.4 log CFU per gram. And the prevalence values were around 2 to 6%. And these values were below the worldwide data. And when we look at the effect of season, spring season as shown in the blue line, yielded with the lowest pathogenic E. coli levels, followed by summer baseline, and winter and autumn, posing the highest risk. The variation in microbial load over different seasons might be influenced by a range of individual and interacting meteorological and environmental events. So to understand the meteorological events, solar radiation, as shown in the red line, and the rain event, as shown in the green line, they were excluded from the baseline model to see what are the results. So when you exclude solar radiation from your model, it yielded with highest load of E. coli values at harvest. On the other hand, the exclusion of rain yielded with the lowest E. coli levels at the harvest for summer conditions. So next, the impact of irrigation water source and manure usage on the level of pathogens in fresh produce was assessed. Again, black dotted line shows the baseline scenario. The usage of tap water resulted in the lowest mean E. coli levels, followed by surface water, yellow line, and the retreated water, red line. So the lowest level of pathogenic E. coli was observed when no manure was used, and this highlights the need for realistic risk assessment in primary production regarding manure usage. The usage of sufficiently treated compost could be an option that risk managers need to discuss to eliminate future foodborne incidences, recalls, or outbreaks linked to fresh produce. So in addition to being a reservoir for foodborne pathogens, irrigation water is also considered to be a potential vector for pathogen transfer from manure-amended soil to fresh produce, depending on the type of the irrigation system used. So the usage of drip irrigation, as shown in the green line, yielded with the lowest level of pathogenic E. coli, followed by furrow and overhead irrigation, which was the baseline scenario. This reduction in pathogen level for drip irrigation is linked to elimination of irrigation splashing. Even though overhead irrigation is more likely to introduce pathogens than other irrigation types, it has been used not only to provide water for produce, but also cool down the produce. So the latter become particularly important for a cool season crop such as lettuce, which is particularly vulnerable to heat stress. For regions where daytime temperature is above the optimal growing temperature, it is recommended to use overhead irrigation only if the water quality is well controlled and potable. In response to recent E. coli outbreak in US, new water safety requirements were put in place in which the usage of untreated surface water for overhead irrigation during the 21 days prior to harvest has been banned. So even though the type of irrigation method chosen by a grower depends on several issues, codes of practices are required to stress the importance of the quality of the irrigation source for ensuring safety of the fresh produce. So the next step was looking at the key risk factors, and sun hours and growing time, they were the most important input factors that were negatively correlated with contamination events. In contrast, the amount of soil transfer to produce by irrigation and number of rainy days were identified as the most important input factors that were positively correlated with the pathogenic E. coli level at harvest. So we published these results in Food Microbiology journal. And if you would like to get a copy of the paper, please reach out to me. I will be happy to provide it. Due to the time limit, I only put the key outputs of the study, but we'll be happy to share more if you are interested. So the development of a risk model using local data, industry data, experimental data, it's a resource intensive but valuable activity as it enables the generation of the realistic estimates of the risk. One thing that is very evident is minimizing microbial load at field is a key through compliance with standards, but we need further research to have more realistic input rather than assumptions, then we can validate appropriateness of the standards. Despite these limitations, the developed risk models remains useful because it doesn't only provide estimates of the risk, but also provides a baseline to compare different scenarios. We hope that this will aid Australian risk managers to mitigate and control risk involving pathogens in primary production. In summary, what I want to emphasize is risk assessment in primary production has assumed a prominent role in industry to facilitate decision-making in risk mitigation. So it's important to be aware of, it's a reliable scientific starting point, not as the final arbiter for decision-making in primary prevention of the foodborne incidences. Thank you for allowing me to share my results, and I will be happy to answer your questions.